Hey, boys and girls. Okay, we're going to try to go a little bit further with teaching you guys a little bit of genetics or something, maybe. Is that presumptuous? I don't know. Probably.
No, it is not.
Okay. So let us review. You remember this?
Yes.
David?
Yep.
Okay.
Yep.
Translation in the nucleus and transcription outside. So transcription in the nucleus, translation outside. I got it backwards. So transcribe and then translate. So basically, and they're talking about, I've been reviewing it's a chapter in a book on information by, I think, Terzis from 2011. And it's actually a paper by two other guys, and they're doing a bit on information in biology. And of course, the whole book is about that. And they were talking about how information is modified inside of the body as the organism develops, and how information interplays in embryology and with the environment and with the genome and, with heredity, and how there are basically those different axes of information that you might look at. But anyway, we'll get a little bit into that. But so anyway, so here we have this, the cell envelope, and here we have the nuclear envelope, and then inside this we have transcription to mRNA, which is kind of a long process. So we might get a little more into how that happens. And then outside of that, we have translation to proteins. And the proteome of any cell usually pretty much 90% describes what that cell is going to be doing, basically. So it's protein content, and every different tissue type in a multicellular animal is going to have a different proteome, a set of proteins that get created in different amounts. 2 cups of this and 1.5 cups of that and 3 cups of this. And that's how the cake is baked, and it becomes a certain cell type. So now where to go from there? Okay. The other thing I pointed out is that how the chromosome is in the cell is a lot different in the last 30 years than what we thought in the 30 years prior to that, basically, how chromosomes existed inside the cell. They used to think it was just like if you made spaghetti and you poured it in the strainer, they used to think it was like that all the chromosomes were all mixed up together, right? And now they realize that chromosomes have territories, because they were able to dye each chromosome a different color with fluorescent markers. And they found out that chromosomes occupy these territories. So you got 2 chromosome 2s, you got 2 chromosome 4s or whatever. And they seem to, and they hang around and attach to this lamin in the nuclear membrane. And now what they don't know is whether or not these lamins have anything special in them that would attach a certain chromosome. So that would mean, maybe this section of lamin has, one kind of cell inner surface to it, and this one has another. And so it would be like more information as to where these chromosomes ended up, but we don't know a lot about that yet. And we're still learning a bit about how the chromosomes are packed in the cell. So, and then let's see, the other thing we went over is, well, no, we're going to go here first. So I'm going to pull up some pictures to make it real. And if you got questions, you should ask questions if you got questions. And there's a good chance that I won't know the answer to all those questions. And
well, the only thing that I'm amazed at is the ability and what we know, and we still can't solve the problem of cancer.
Oh yeah, well, there's a really good reason for that. Solving the problem of cancer is almost like solving the problem of biology.
Yeah,
because it's that big. I used to think like cancer's got to be like one thing, right? One thing that happens, and why can't we find that thing and fix it? Well, it turns out, like every week we discover new things about the cell, and every week we find new cancers, and new situations. So it's going to be a matter of learning how to make designer drugs for very, very particular individual cancers and individual human beings. And we are getting there faster than, I'm hoping faster than I think, because a lot of money is going into that sort of thing. So anyway.
Yeah.
Okay. So you've got the double strand. This is a picture showing right here of the double strand. No, this is not a weather report. Not a weather report. Nobody's getting in here. Weather report, we let anybody in the house. No one's getting in tonight until maybe later if I weekend, but not even then probably. Okay, so let's take that off the screen. Okay, so we got a picture of the molecules here, right? And what do these look like to you guys? I don't know. I mean, you see oxygen atoms and phosphorus atoms, right?
Yep.
And stuff. Okay, so this is the ribose backbone that you're looking at in black and white. Going and you can't see my cursor, but basically with those little phosphate groups linking those riboses together, and then in between is the actual nucleotide, just guanine, cytosine, adenine, and thymine. And those things complement each other, so C will attach to G and A to T all the time. And a good way to remember that is GICTA, or you can yell it like a cowboy, GICTA or whatever. Mm-hmm. And and so it's GCTA. So that's the natural way that it goes. So this is a double-stranded DNA. So this would be one chromosome. And it would be, say, the chromosome that you got from your mama. So chromosome 2 maternal, basically. And then you'd have another one that was almost identical to it, but not quite from your daddy. So you got two of those. And they're in those nuclear regions, right? And so this is the thing that's going to give us the code. But now from there, DNA tends to, let's see, here's a picture. Yeah, this is a picture of mitochondrial DNA and basically the nucleus. That's not a good picture. This is a good picture of how DNA packs up. Remember I told you about those?
Yeah.
Remember I told you about, like normally we thought of chromosomes as those X's on the right?
Yeah.
That's the way they are when the cell is dividing, or when you're creating mitosis, or when you're creating little eggs, little baby chromosomes, meiosis. So those two different things. It ends up looking like that, but normally it's, it is really packed up. And if you look at those arms, the arms of the thing of the X, basically, that's what it's showing you here, how it's completely packed up. You've got the single chain going in, wrapping around the histones, and then it wraps again, and it goes into a bigger structure. And it's amazing that there's a few things about this structure that we don't know yet, which kind of surprised me. But here's the different levels, and let me see is there any way I can make that bigger? I'll try to make that a little bit bigger for you without getting rid of my thumbnails, which I don't want to do right now. But okay, so from now, this is the double-stranded DNA on the left, and then it wraps around the histone, and then it kind of folds into, it's wrapping around a histone, so that's called a nucleosome when it wraps around a histone, and then from there it wraps into a further structure. And then it wraps into this. They measure them by how many nanometers they are, and then it folds into that, and then it crisscrosses here, and then it folds up some more, and eventually you end up with that. So the point here is that DNA, when it's packed like that, there's no way that it can be transcribed into RNAs and mRNAs. A little bit of it could, but it would it basically run into other molecules, and it'd have a big problem. So when it gets into when the cell gets into its growth phase or its normal phase, the DNA starts to become unpacked for various reasons, and how it unpacks determines what the cell type is. So,
okay,
heterozygous. What? Oh, heterozygous will be both copies of the homozygous. Okay, well, okay, I'm not sure, Creole. If DNA is okay, because you misspelled things so badly. DNA is heterozygous. Will both copies homozygous with different variants? Yeah, both copies, both of the double strands will be you could say they're homozygous with each other. They have to be because they bind together. The two single strands that bind into the double strand that form one chromosome. Okay, but it could be different from the other chromosome. So. And let's see. So, how we doing, guys?
Good. Keep going.
Yeah, keep going. I'm doing fine. I'm trying to stay awake, but not because of the topic, but because of the hour.
Oh, that's right. You haven't got you barely got any sleep. So, Orge, your mom wrote the code. Okay, that's the answer. Your mama. Answer. Your mama. Your mama did it. Okay. So then let's see. We might have some other pictures here. Nope, that's something else. Another subject. Sorry. Okay. Yeah, here's another picture of it coiling. Basically, this the double-stranded wrapping around the histones, which have 4 different proteins and 2 copies of each. So it's an octet. So it's an octamer. And then it folds up into another structure, another structure, another structure. So we got to get that unfolded in order for something to happen. So that's the point. Okay.
Wow, this is really complicated.
Yeah. So
really,
what are we missing?
What are we missing? I don't know.
What are you missing?
You're going to have to tell us, teacher.
Yeah, I'm
supposed to tell you what part is complicated.
Yeah. Okay, I'm not going to become a microbiologist, and it was so far probably an hour and 15 minutes. Or
look, out of all of my 432 subscribers, you two are the ones I picked because you two are the ones that are most likely to become microbiologists in 48 hours.
Yeah.
Well, that's
if I could accomplish that, boy, that would be pretty amazing. But I don't see that.
We got another question from Creole here. So watching the questions in the chat is something we got to do too. Okay, if I don't want to miss any. Okay, so it is in the organism. Some cells will express one trait and some cells the other. Yes. Yeah. And the reason for that is a little bit complicated. Okay. We've got these territories here. Okay.
You're in trouble now.
What? Oh shit. Oh no.
Yeah.
Missing chromosome territory. What? I'm missing some?
The big teacher's here. You're in deep shit.
Yeah, maybe he should come in and help us out. I could send you a link, Demeter. I could send you a link. Anyway, so
come in, Demeter, if you want. Go ahead.
The question I don't think we know all the details about how this stuff unwinds. We're learning more and more. We know that some of the ends end up like all collecting together in something called the nucleolus. That there's, which doesn't have a membrane around it. It just happens to be a region, like a little tangled knot at the center of the nucleus, and that's what creates all of your ribosomes and some of the more primitive characteristics of the cell. Basically, they all seem to gather in this little nugget. But anyway, so for whatever reasons, these strands, through histone modification, through epigenetics, some of these things become a little bit unwound. So there's some slack in different areas, and there's some interaction between and within chromosomes that manage to pull out some of the loops and fold them together so that they can transcribe. So the main point is the simplification here. The only thing you need to remember is that there will be some modification of this tangled web of spaghetti so that some of the spaghetti is sticking out there so it can be transcribed easier than others, and it will be transcribed. And there's also, as Demeter reminded me last time, these histones, where you have 192 DNAs wrap around, and then you got space between the histones, and these histones can kind of slide along that string at different points depending on histone modification. Tangelo's always talking about those 34 epigenetic codes or whatever the hell he's got.
Yeah.
I forget how high he keeps adding one every week, but he's talking about all the different ways that this DNA, that this tangle of spaghetti can be modified. So if you had a strainer full of spaghetti and you grab one noodle and you start pulling it out and stringing it out a little bit, okay, you can basically copy the secret code from that noodle onto another noodle. A different kind of noodle, because it would be an RNA noodle, right? So you go from DNA to RNA, which is very, very similar but different.
Yep.
Have I confused? Okay. So
no, I'm really thinking hard to try to come up with some good questions, but I haven't really. Okay. But keep going.
Okay. Did I get into?
I'm usually pretty good at that, but
yeah, I'm trying to see if. Okay, what did I do here? Idiot dead. God did this. Multi-game worldview. I don't know what's going on.
Yeah, that could be some of your old stuff, right?
Yeah. Stuff's not erasing well for me today, or whatever. So anyway, the whole point is that you're going to end up with different cell types depending on how the spaghetti is sitting in the strainer. As to what you can transcribe. That glob of spaghetti that's at the center that's all twisted up, you're probably going to have trouble transcribing that. So anyway, I'm not sure what he meant by missing chromosome territories. Are you complaining because I didn't put all 46 of them in there? You better not, because I'm going to call you Danny. Pedantic. Whatever. Anyway, okay. So where do we go from there? So now there's other things. So it's okay. We got a ball of spaghetti up here, right? And it's basically unwrapped and it's looped around itself because there's a piece here that somehow tends to attract itself, and we've got a little strand of DNA here that we're going to transcribe. So we're looking to transcribe this little piece of DNA, which is going to create a protein for ourselves. So now we're looking inside the nucleus, and we're looking into how it gets transcribed. And we will select that color. And so this little section right here is what we're interested in, right?
Yeah.
It's got a bunch of things in it called exons. And exons you can think of as they get exported, and introns just stay inside the cell and we throw them away, basically. So we've got a gene, basically right here. This is not a double-stranded thing. Let's think single-stranded for a second. And inside of this gene, we have basically these exons. And in bacteria, they don't have these exons. They're just the whole thing gets read as, one big strand of protein. Except for, I think there are exceptions, but the exceptions are pretty rare. Anyway, so we got this part here that we're interested in, and here we have this. And the thing is that there's a whole lot of intron for exons, and I can show you a picture in a database that'll show you that there ain't a hell of a lot here as far as exons go. So, okay, so further, in order to transcribe that thing, this is called upstream, going to the left. It's called up the stream. So the river's flowing this way, right? We're going to be transcribing this way, basically left to right. And so upstream, there is a spot right in front of the gene called a promoter. And oftentimes it is called a Tata box, not to be confused with the consulting company in India called Tata, oddly enough. And that is a thymine, adenine. And what happens is, am I going too fast?
No.
No, I was just trying to make a joke about typology.
Hang on a second.
What was that?
Sister calling. I have her as that robot voice. So I'm going to have to send her a message. Hang on. She's going to get pissed at me. My family is so mad at me for being online. They claim they can't talk to me anymore and I don't exist and I've abandoned them.
Oh, yeah.
Yeah.
You getting the cold shoulder because of it? Yeah.
No, a little bit.
Yeah.
A little bit. Okay. So anyway, so a protein complex has to attach to this thing, that has a couple of different parts. And it basically kicks off this whole transcription process so that it can start reading right here and read this entire strand. And it reads the exons and the introns, and it creates this pre-mRNA.
Yeah.
Okay, so it gets this whole thing. The next thing that happens is we gotta splice out this part. We gotta splice out these introns, and each of these introns has a little signal in there that tells it how to splice. And what we end up with is mRNA. Whoops, gotta be able to write with something.
So
Let's see.
I have kind of a dumb question.
Oh, good.
How did science actually learn all this? All through a microscope?
Oh, shit.
Basically? Or
they started with a microscope, and then they used x-rays, and then they, for a long time, they would freeze the cells, and they would freeze pieces of them, and they would x-ray them and check them out. Now they've got a system where they can actually watch them in real time. And then they have special little pieces of DNA that will create a fluorescent protein, so they can look at it under fluorescent light and see where certain things are. And then for every protein structure, first of all, like for this right here, this little glob that helps you transcribe, they first of all had to sequence it, right, to figure out what amino acids are in it. And then they had to go find it. They had to go find it in the genome, and which is kind of hard because there's a lot of redundancy going from, there's a lot of, there's more than one codon that will hook up to each amino acid. So you got to have a program that'll go and try to fit that protein to the genome. So then they know where it came from. Okay. And then the next thing, they have to crystallize this thing. And not all proteins crystallize. And this, Demeter would know a hell of a lot more about me because it's actual lab work, I call people that do that stuff lab rats. I probably shouldn't call them that. But anyway, so it and it's just when you look at some of the real geniuses that came up with new methods, and they're coming up with new methods every day, and there are many geniuses out there, they go through just this horrible pain to find things, right? They use chromatography, which is where, like, basically if you had three colors mixed together of different chemicals, and you had them all mixed together in a liquid, and you put it on a paper towel, like a Bounty paper towel, it would separate out as it went down the towel. You might get the red dye on top and the blue dye and then the orange dye. So it would separate them out. So they use that. Then they have electrophoresis, which is like an electrical way to do chromatography, and it is incredibly complicated how they figure all this out. And that's why we don't know everything about how those chromosomes are arranged or what they're doing. For one thing, they're moving really fast. The shit's flying around in there, flopping around, and we got to try to slow it up and capture each individual part of it. And cloning, RT transcriptase, lowest dimension sequencing. He's offering some ideas. Anyway.
Mm-hmm.
So what happens after splicing? Oh, shit.
But a lot of this stuff, would a lot of it be learned with computer models and things, I would assume, too, right?
We're getting there to the point where we're doing more and more modeling.
Yeah.
But that's where you get down into, what is the information and what's it doing and stuff. But to computer model what's going on inside of one nucleus and one chromosome and one nucleus is probably beyond our computational power right now. Even if we were to commandeer Amazon, we'd have a hell of a time with some of these models. Okay, when you're done splicing, you end up with just the introns, right? And whoops. Damn, stop it. So we got a little short intron here, got a longer intron, got a medium length intron, another short one, another long one, and we got a couple more. So you just end up with the introns. So now we got some good mRNA. And then this gets further processed. Got some stuff added here, and we got some stuff added here to basically protect that mRNA so it doesn't get degraded. And then it goes out of the nucleus, and now we can hit the ribosome, and we can make ourselves a protein. So that's kind of the whole story about how a protein's made, right?
Yep.
Okay. So now there's dozens of different methods of untangling that DNA, and then there are other things. There are promoters, and then there are upstream enhancers and repressors. So up here you could have either an enhancer or a repressor, and these things tend to loop the DNA a little bit. And they attach to other regulatory proteins, so that now we have the situation of, okay, we got our proteome, which is a set of proteins, right?
Yeah.
And that's what's in that proteome can be modified by these enhancers and repressors and many other mechanisms. And the other thing it can be enhanced by is methylation. Okay. So, and you've heard about that, right? With methylation, I think that was the first bit of epigenetics that we found, is that these things here, the cytosines, when they're in, they're in a CPG island. So basically, they're in an island. They're close together, so we have a C and we have a G together. Okay, this cytosine gets a methyl group attached to it, which is a carbon and 3 hydrogens. And when that happens, it represses that gene, so it shuts it off. Okay. Well, then the next thing they found out is that when mama is starved, that big experiment they did in where the Nazis basically prevented people from getting food and starved them. They ended up with DNA methylation that actually got transferred in the eggs. And I think it was eggs more so than sperm, but I think sperm happens too. So they found out that this epigenetics can be transferred to 2 or 3 generations downstream before it becomes unmethylated. Now, usually what happens is when the egg is created to go from one organism to another for you to have offspring, all of the methylation gets stripped out. A lot of it does. And all of the epigenetic effects are pretty much stripped out so that you don't transfer too much from one generation to the other. But then they found out, oh my God, we were so surprised. You can transfer some of that information. So, and now I'm going to tell you how I prove that information arises naturally. But are we ready for that? So, okay, and anything I've said in the last few minutes that is incomprehensible?
No, I don't think so. I'm just sitting here in wonder, amazement, I guess maybe another way to describe it, on how much that you know and how much you've studied for a person that is not actually doing it for a living, and how much you know about this stuff and what you've taught yourself. So that's kind of what's gone through my mind for the past
and what
minute,
really kills me,
the past minute, basically. Yeah.
What kills me is how much I don't know.
Yeah. Well, I'm sure. Yeah. Well, look at how complicated it is. I mean, that's nothing to be ashamed of.
Yeah.
You know, I mean, it really isn't. I mean, you know, no matter how much you learn, there's going to be tons more, right? No, I mean, it doesn't matter how far you get. You're never going to see the end of the tunnel because of how complicated it is. So certainly don't let that
hopefully I got
deter you, or don't let that deter you, or bother you. You know, so.
I've only been hitting it for about 3 years.
Yeah,
and prior to that, it was philosophy, mathematics, and neuroscience.
Yeah,
and I did have a lot of molecular biology background prior to that, and then chemistry before that. But I mean, this, you can put together pretty good knowledge if you just stick to it and keep attacking and figuring out.
Yeah.
Like I would sit around and think, oh God, transcription, how the hell does that happen? I have no idea. And then go start looking at the books and figure it out. So anyway.
Yeah.
Okay.
Well, you had like you just said, you had at least a decent background, and to get you started anyway.
Yeah, the chemistry part is really important.
Yeah, that
we might divert to that yet tonight. If you guys feel that your brains are not sufficiently.
I mean, at least dive in a little bit. That would probably be good.
We could do a little bit. But first of all, I want to show you. Okay, so we got the mama. Here's the mama. Okay, and the mama. This is the mama's nucleus, whatever, right? And because of the environment, because of the effect from the environment, right? There is information in the environment, and that information gets transferred to mama's nucleus in the form of methylation, of epigenetic modification of that DNA, right?
Yeah.
And lo and behold, mama passes it on to baby. Baby's very sad because baby hasn't eaten yet. Babies are always fucking sad, right? Baby doesn't have much hair. Baby doesn't have much hair either. So okay, so we've got information coming from the environment, telling you that the environment is harsh and bad, and you have to conserve calories, right? And that information gets encoded onto mama's DNA in the form of a methylation pattern that happens, somehow, biologically, it'll create a pattern, encode that information on that DNA. That information gets passed to the baby, and then baby becomes happy because he gets a girlfriend, and pass it on to another baby who's also extremely pissed off.
Yeah.
Okay. So now we got the information passing on 2 different generations. Where the fuck did that information come from?
Right. Now here's where the creationists will really give you. Yeah, exactly.
Yeah, but how will they? Because it's obviously, we know that this is all, they are not going to say that a miracle occurred here. They're going to claim that it was all biology, right?
Yeah.
We have for 3 generations. There is no miracle here. No miracle.
Yeah.
So we have information transferred from the environment onto a genome, passed down to the children. Okay, but then it kind of dead ends right here, because the chances are that it'll become unmethylated, and it will go from there. So they would say, "Oh well, so epigenetic." But what they're claiming now, standing for truth, is kind of saying in his lectures that epigenetics is enough, but we know it's not. We know it isn't a really good method to get information into the next generation. But in this case, from here to here to here, that is a natural creation of new information that gets inherited for a couple generations. And we know that, and we've proven it, and we know it's completely natural. So I don't know how you could push back on that.
And why does it stop? You said it stops there? Like,
I'm not... Okay, I'm not a specialist in inheritance and epigenetics, and I don't think science. I don't think science is right now. So
right.
But there's got to be a ton of papers out there. Now, I did read somewhere that fungi, or fungi, whichever way you want to call it, that they are capable in some cases of passing it down as much as 100 generations. So they found epigenetic modifications 100 generations.
Probably certain things can go further than others, right? Certain information. Right. Yep.
Yeah. So that would be a case of basically information from the environment going down 100 generations for mushrooms.
Right.
So, and there's just a lot we don't know about those epigenetic modifications.
Is there a lot of study in that area, or?
Oh yeah, it's probably going wild. There's probably 20 papers out there in the last month, that we
right. And
now here's what Creo Debunk has nailed the next thing that I was going to talk about. Basically, they want to know how the mutation information arises naturally, and the answer is again from the environment, right? And he's correct about that. So now here's the other thing: is that when we end up with these copies. Oh God, this thing's way too sensitive to my hand.
And all of this stuff is a perfect example of what nature sucks at it, right? That would be a kind of a way to describe it, because of, like you said, it only goes like certain things that are inherited only go a certain amount of generations. It may, other things do. So you end up with extinctions, right? I mean, kind of like. It just sucks at it. So that's why there's so many problems, and that's why there's so many mutations and different things that, okay, keep going.
Yeah, before we go there, I want to point out that, let's see, where did I do that process? I did it up here. Okay. The thing I was talking about is this regulatory area here where things are regulated. Okay. Okay, a very, very, and this is something Andrew Cumming has talked about a lot, but if there's a very small change, like that Tata box gets modified, lose one of those.
Yeah.
That very small change can end up modifying how much protein is in this proteome for a cell while it's developing. And you could end up with an animal with a much longer humerus than its ancestors. Just from that one modification, right?
Yeah.
So you could end up with, or you could end up with another bony ridge that a muscle attaches to.
Yeah.
That the other things don't have. Like if you compare human beings, we have a difference in where our biceps attach that allow people with much smaller muscles to lift heavier weights. Did you know that?
Yeah.
It's just leverage. If it attaches a quarter of an inch further or closer, you get a tremendous amount of leverage, and you can lift 10 pounds more with the same muscle mass as the guy next to you. So we have a change right there. So very tiny mutations can make very big changes.
Yeah.
And that was one of the things that they emphasized the hell out of in this book that I'm reading. So. And anyway, I just wanted to point that out. So now we go to the point where every time we go through one of these generations, we end up with mutations, So
Yep.
we have a mutation here, mutation there.
Yep.
And then the interesting thing is that the environment.
Okay. Yeah, I was just about to bring that up. Yeah, go ahead.
The
Cut
information
that right off, right? I mean, the
Yeah, the
environment something can
information-
happen. Yep.
in the environment can do one or two things or three things, basically. It can reduce, can be deleterious, or it can. What the hell
Yeah.
happened here?
That was
Stop it. Stop it. Damn thing.
yeah, can be beneficial or not. Yep.
Can do absolutely nothing. So neutral.
Yep.
Or it can have some small benefit, like you can lift a heavier weight.
Right. Yep.
And if it turns out that lifting weights, the environment as we're going forward, it becomes more and more important to lift weights. And these guys here are going to end up in the future. And we're going to have people with biceps that are attached, an inch further than the other ones. And you're going to end up with a morphological difference. So at every point here, we have information being transferred from the environment and filtering. And this is natural selection that Danny says we should do away with. This is the important part. This is where the environment imprints the information on the organism. And that is really important. And what they don't want to believe, you either want to believe this shit or you don't, right?
Yeah. Yeah.
Like in 6,000 years, okay, at one generation every 20 years, we end up with, we got 60 times 5, we have 300 generations, right? Okay.
Hey, Speed.
Yeah.
Creole wants to know the name of that book again. Is it in the description?
I will put it in the description, and I will pop it up here in a minute because I managed to get a PDF copy of it. I've got a bunch of highlights in the actual book. Let me find it here.
Our friend is here too.
Oh, LPV.
Yeah.
He's laughing his ass off because he didn't believe any of this stuff. Come in here, John, and tell me where I'm wrong.
Yeah.
Okay. Yeah. So we have 300 generations, but what do we got in 4 billion years? And considering we're talking about human generation time, which is 20, if you're talking about bacterial generation time, you're looking more at like at least one a week. So 50 per year times 4 billion years. So we have 200 billion generations. So how much bigger is 200 billion than 300?
Yeah.
5 enough zeros? Am I doing it right? Yep. Okay. So 200 billion generations with those very small changes the whole time. You've got a changing environment.
Right. Yep.
Passing information, imprinting information on that genome all the way through.
Yeah.
And so you end up with this accumulation of information, and that's how it's created. And it's done with that filter, the environmental filter, the very fact that the things that end up staying alive are the things that have the right fit for this environment.
Right. Yeah.
Which is Danny's tautology, by the way. So anyway, you talk about this all the time, John, but do you really know what the hell you're talking about? Can you come in and tell me how I'm wrong? If you want to come in, I'll send you a link. I think I have your email yet. So anyway, but the first thing you have to buy is that the Earth isn't 6,000 years old. If you believe that, you're pretty much fucking gone, right?
Yeah.
Because there's not enough time for this kind of information to be accumulated. Because this information accumulates very slowly when you consider that, we're looking at I guess the next thing we'll talk about in the next episode, we call them episodes, is the different kinds of mutation and how proteins have this remarkable ability to work like Legos, and how all of that so-called junk DNA ends up being what drives evolution. It ends up being like your garage full of really good stuff that you hardly ever use, but when you need to use it, you go dig through stuff, you cobble something together, and you got a new thing, and that happens a lot. So. Okay,
Yeah, we should get into a little bit of the chemistry too.
we'll try that, but
Yeah.
very, very, very light, very light.
Yep. Yep. That's a mutables kind of specialty, right? He's more
Yep.
on the chemistry part of it. Yeah.
Yeah. So the most important thing is carbon. Okay, so we got a carbon atom, and carbon has 4 little hookups, so we can always hook to 4 things. Okay,
Yep.
and if these 4 things are hydrogen, then we have methane, which we all know about. I guess it's all all over the place, right?
Yep.
So that's the first part of it. And what's cooler is that carbon could hook to another carbon like that, and then hook to 2 hydrogens.
Yeah.
And hook up like that.
Like that. Yeah.
And if we were to draw that, we would say CH3, 1, 2, 3, CH3.
Yeah.
Or if we were to write it in shorthand, basically. Okay. That sort of thing. And if, so that is ethane, the one I just drew, right? So we had methane, ethane, and now if we hook up to another carbon, like that, so now we have a CH2 in here, that's propane. Okay?
Yep.
And now if we hook up another one, what do you think we'll get?
Is that a Minnesota way to spell propane? Oh, that's a little squiggly mark, right? So you just...
I forgot to complete it.
Okay, all right.
Okay, fucking pedantic fucking kids today, man. Anyway, okay, that's butane, which you have probably lit a cigarette with or a joint, right? Butane lighters?
Yeah.
Okay.
I have used many a butane lighter on things.
Yes. Okay, good. I'm glad. Okay, I just want to check, see if you guys are with me.
I'm with you.
So methane, ethane, propane, butane. So, that's with 4 carbons. So these carbons, and the neat thing about these carbons is they can link up in, they can also link up to other things, like an oxygen. Okay. So now we got, and we're not, I don't think we should draw the hydrogens. We'll just do this. So just go like that. And we'll pretend there are hydrogens there, okay? Because we're going to get sick to death of drawing hydrogens. Except this oxygen wants to hook up to 2 things, so it's going to have a hydrogen, okay? But first of all, we only have 1 carbon. And, okay, so this one, we're going to put the hydrogens there. Okay, this is methyl alcohol. Okay, this is called a hydroxyl group or an alcohol group. And so this would be methyl alcohol, which is wood alcohol, which hobos drink, but you should never drink it, kids. Okay, because it'll make you blind. That's methyl alcohol. Does that make sense?
Yep.
That does make sense.
I'm glad I'm making sense because I'm getting tired. What if we put 2 hydrogens? You remember we had like an ethane, right? With 2 hydrogens. And then we have an O and we have an H. What do you got? You guys recognize that?
Cheap gasoline?
Ethane or ethanol.
Ethanol, yeah.
Ethanol. Remember ethane? Okay, methyl alcohol, methane, methyl alcohol, ethane, ethyl alcohol, propane, propyl alcohol. So, and I want to show you something cool. But first of all, we'll do propyl alcohol like that. So we have an OH there and hydrogens here. And I said I was going to stop drawing hydrogens. But what's really cool is if you add another one and you have butyl alcohol from butane, right? Okay. With an OH. So we have 4. That's called butyl alcohol. And that's actually a byproduct that will give you a hangover. And there are other higher alcohols, like the higher alcohols that yeast forms is often the stuff called fusel oil. It gives you a hangover. But there's one other interesting thing here, which is going a little bit further, okay, and that is that this thing right here, where an oxygen is double bonded to a carbon, okay, shit, where am I? A little eraser there, okay. This is called a carboxyl group, but this stuff here is called butyric acid. So it comes from butyl alcohol, right? And it got its name because when you smell rancid butter, it has butyric acid in it. And that's where butane got its name from butyric acid. So, isn't that cool?
Yep.
I think it's cool. Anyway, this is stuff that really took me in my youth and fascinated me. And I read most of it in an Isaac Asimov book called The World of Carbon, which really got me going on all this stuff. But anyway, that's butyric acid.
So it was a nonfiction book, or did he like do a sci-fi on carbon?
He wrote a lot of nonfiction books. Did you know that?
Yeah, he was prolific.
He was prolific as hell.
Yeah, he wrote what, over 300 books or more than that?
I think so. Yeah.
Yeah.
I'd heard that he wrote like 30. What the hell did I do? I heard he wrote like 30,000 or 30,000 words in one day or something like that. I don't know where my drawing just went. Okay, there it went. That sort of thing. So we're not going to get too far into the chemistry here, but this is just, the structural aspects of chemistry. Okay. And now you've heard of isomers, right? So we got a 3-carbon chain, which we had in propane. Okay. So that was propane. And we put a alcohol here and we made propanol. Okay. But if instead when these carbon chains get longer, they become more and more ways to vary them. So if we had a hydrogen there instead of an OH group, and we had the OH group right here, we would have isopropyl alcohol, which was everybody's friend during the pandemic. Okay?
Yep.
And the other one is propyl alcohol. So, and then there's isobutyl alcohol, and there's so anyway, the exercise that you should think about is let me just try to. I wish I had a big eraser, like a big fat one. That would be cool, but they don't because they suck.
You can't set the size of your eraser in that app?
Yeah, you can. Let me see if I got it on Max. I think I guess I maybe didn't have it on Max. Anyway, you set it a little bit. Okay. The thing is, you can hook these carbon chains up in a number of different ways. I mean, you can just keep going. You can have a carbon out here and a carbon here. And you can have a oxygen there or another carbon there and branching like that. So anyway, that's like one interesting thing about gasoline is something called octane. I think I always get it backwards. I think octane is the very best burning petroleum product. 1, 2, 3, 4. So it has 8 carbons in a straight chain, right? Like that. I kind of screwed that one up. Yeah, now I'm going to erase a whole bunch of shit because I have a big eraser now. So I got 6. So you got 8 carbons. Okay, that's called octane for the octane rating. And I think that one burns I think this is the one that burns really well. It burns really well in your pistons. But if you got, like, this is 1, 2, 3, 4, 5, 6, 7, 8. This is also octane. Same thing. It's an isomer, right? This one will make your car knock because of the structure. So it's apparently, and it may be just the opposite, of which somebody in chat knew. So anyway, they call propanol a primary alcohol and isopropanol a secondary alcohol. Anyway, so these are both octane. And gasoline usually comes in this form here, where it's all screwed up. Basically, it's all, interlinked, because nature is messy, right? And you do an octane rating by comparing to how well this burns, I think, or vice versa. I can't, but yeah, I'm pretty sure that's the octane rating. That burns the best. So if you can get a hold of pure octane, I think your car would burn better, that sort of thing. So,
Yep.
Okay. So anyway, the point is that you got to think in terms of how many different ways you can arrange these carbons. And there's something else you can do with them, which is even cooler. You can go like this. 1, 2, 3. You can make rings. So anyway, it just goes around and around, and it hooks up to a ring. And that is what the hell is that called? Oh shit, I forgot. This is benzene, basically. If you have double bonds, so then you only have one. Okay, every carbon has to have 4 things hooked up. You can make double bonds between these things, and so you would have, let's see, hydrogen there and a hydrogen there, and so each of these would have a hydrogen hooked up to them, right? Because they all have 4 bonds, that sort of thing. Okay, yep. So you can hook them up in rings, and that's what you're seeing when you look at those diagrams of the nucleotides. You're seeing something a little more complicated.
You got it right. Mutable.
Good.
Mutable making coolness. Yep.
But you can do other things like, let's see here. 1, 2, 3, 4, 3, 4. You can put an N in there, right? So it can hook up to a nitrogen.
Yep.
Along with a carbon, or it can hook up to an oxygen. So there are, these are called heterocyclic compounds, and it becomes more complicated. So anyway, what I want you to take away from that is only 2 things: that this is a hydroxyl group, and it is an alcohol, and that's the first little group that you learn, or that you can learn, hydroxyl group, and it's an alcohol. And the other thing is that you can hook these carbon things up to incredible, like, varying chains of things, right?
Yep.
And that is the complexity that life is founded on. The number of different ways that you can hook these Lego blocks up.
Right. Yeah. Yep.
Very different compounds, sort of. Yes. Oh, cyclohexane. Okay. Sorry. Yep. Been a while. And then we could also hook a OH up to that. We can make that an alcohol. So when you go and look at the wiki and you see these structures, just look at them as, just look at the atoms that are there and imagine how they're bonded. And remember that carbon wants 4 things hooked up to it. Nitrogen typically wants 3 things hooked up to it. Oxygen wants 2 things hooked up to it. And with those 3 elements, you can produce an amazing complexity of compounds, which we could get into.
Yeah.
The neat thing about these guys is they're called aromatics, because when you hook them in chains like this, they do a weird thing called resonance, where this will be like sort of this double bond will kind of float around the circle. So it goes around and around. And they are the basis of a lot of things that we consider good sense or scent. How we smell things. A lot of them have aromatics. So a lot of these ring structures have a great aroma to them.
Yep.
So you can come up with some examples, but I think that's probably enough. So, yep. And the resonance is a very important factor that makes a big difference in the reactivity compared to what it would be if it didn't do that, basically. Yeah. There's a guy named Kekuo, I think, that famously was on a bus or something, or driving or going somewhere, and he fell asleep, and he dreamt of these monkeys in a circle hooking their hands together, and he saw this resonance thing in his dream, and we don't know if that really happened or not. It may have, may not have.
It's also
Okay.
the origin story of that barrel of monkeys that they used to sell to kids, little monkeys that would look together.
Yeah, exactly. Yeah. Yep.
I forget what they were called. I think it's called barrel of monkeys, but I could be wrong.
So
It's been so
apparently,
long.
apparently, Scott, me and you have little pee pissed off because he claims that we don't know shit about this stuff, but we talk mad shit about him.
Oh, sorry about that.
Yeah,
Well,
Must be tough.
yeah, must be rough.
you know,
I talk
it
mad
isn't
shit
hard.
about you, John, because you're a friggin' dipshit. That's why.
Yeah, it isn't
It has
hard.
nothing to do.
You know, you guys are very good judges of character, better than me.
Yeah,
So-
it's right. That's what it's about. It has nothing to do with biology or chemistry or anything, John. Well, it might have something to do with. Well, I guess it might have something to do with it that because yours is all screwed up because you're
Yep.
a dipshit. But other than that, it has nothing to do with the knowledge part of it, whether how much you know or how much I know. It has nothing to do with that.
Yeah, and I bet you Maddox couldn't have drawn the structure of alcohol, ethyl alcohol.
No.
He can now
Yeah.
because I just taught him,
Right.
right? Or he can Google it fucking faster than shit, you know. But he doesn't know crap about chemistry either.
Yeah, that's what's cool about the internet is you can learn all sorts of different things at all sorts of different levels.
Yeah, methanol, ethanol, propanol, butanol, methane, ethane, propane, butane,
Yeah.
pentane, hexane, heptane, octane, nonane, undecane, or is it undecane? I don't know. These and other questions will be answered in a future broadcast. Okay.
Okie doke, man.
So I don't know. Questions? Is there anything that's overwhelming?
Nothing, not overwhelming.
Okay.
Eventually, I'm gonna have to read up this stuff on my own.
Yeah, I think the thing to think about is transcription, and then translation, and once it goes outside the nucleus, and then it's translation to a protein, and then go back into the nucleus and start thinking about introns and exons. Exons are exported to the protein. Introns are spliced out. And then think about how a gene is turned on or off, either through epigenetic modification or through a series of regulators or inhibitors or enhancers or whatever. And you pretty much have the whole story in a teacup, you know? From there, it's just a matter of putting the flesh on. So, I think. And then go to the wiki and look up ethanol, look up methanol, look up a carboxylic acid, and start just looking. Instead, like normally what a person does, I think what an average person does when they see a chemical formula is they go, oh, gobbledygook. Don't gobbledygook again. Look at every single molecule and look at the shape. And if your doctor prescribes something to you, go out and look at that son of a bitch on the wiki and see what it looks like, what its structure looks like. And watch out for those fucking chlorine atoms because they're constantly sticking them in our drugs. I think it's a chlorine conspiracy.
Yeah.
Oh, by the way, we were on a walk last night on a path in Chaska, Minnesota to go see fireworks. All three of us had the same Pfizer vaccination. When we walked down that path, it was pitch dark until we passed a certain point and a light came on. And then we passed another point and that light came on and another light came on.
So you think your microchip turned it on?
Yes.
Yeah.
Yep.
Wouldn't
Could have
have
been magic.
anything to do with a motion sensor or anything like that on the light. Wouldn't have anything to do with that, right?
I don't know what you mean.
It was just a chip in you. Yeah, you don't know. Yeah. Okay, Danny. Sorry.
Okay. Okay, guys, I think we should wrap it up.
Yep. It was fun.
Unless I mean,
Unless
if
John
people,
wants to come in and educate us. John can come in and educate us if he wants.
yeah,
I mean, he's so brilliant. That's what amazes me. I'm more amazed at that, really, than anything else.
we are amazed at his brilliance.
Yeah, it's just... Yeah, yeah.
Well, the interesting thing is, by the time his brilliance comes out of his context in delineating and tortured sentences, it doesn't look brilliant anymore. So that is the most brilliant codification of knowledge I've ever seen, because we can't tell that you know anything, John.
Yeah.
By the time you spit it out. Okay.
I'm gonna get a hold of YouTube and see if they can give out a Nobel Prize for John, maybe, or
For
a YouTube Nobel Prize or something.
spaghetti sentences.
Yeah, spaghetti sentences. Yeah.
Oh, the other takeaway is think of the masterful DNA that is in your nucleus as a big ball of spaghetti.
Yeah.
Because that's kind of biology amazingly works like a big ball of spaghetti. Oh, I was going to show something else. Hang on a second here.
And one thing I enjoy about doing this is I hope I irritate the shit out of you, John. Because it's my pleasure. That's exactly what I'm here for. Just to irritate you.
We enjoy the hell out of that.
Yeah, definitely.
I was going to do one more thing before I left. And that is go all the way to the top of this document, and hopefully there aren't a million papers up there. And okay. This is the book. I'll put it in the description too. Philosophical and Scientific Perspectives by Terzis. And let me go check the page number. Where the fuck am I? 203. It's around 203.
If we on our next one that we do of this, we should invite Demeter and see if he obviously he loves talking about this. So
yep,
should ask him if he wants to join us.
Let's see the article that I don't have any highlights on this, but the self-construction of a living organism, and this is all about information. And we could go a little further into this in some future talk, because they do some pretty neat things here. Let me see if I can figure out where they do that neat thing. Right here, where basically they do a little bit of math. And this isn't real math. It's like modeling math, like mathematical models.
Yeah.
And they talk about basically this T1 here is the time. GI, which stays constant, that's why it's capitalized, is your DNA. And the small GI is your epigenetically modified DNA. And somewhere here they have like the epigenesis thesis, and they talk about information and how it's transferred from the environment. So, John, if you don't believe me about the information, this article here will set you free. And it's written by some of the same guys that were active in your March 2016 Royal Society group of information papers that you like. And these guys are referenced in it.
John just likes working off of his imagination. That's all he wants. His
little
imaginary friend that created all this. He prefers that. Yeah.
That's a pretty intense little paper, and it creates a nice little model for information in biological systems, which I like. And nowhere in here do they say anything about God doing it or about the impossibility of it happening naturally. In fact, they're where I got the idea for the title of this episode. So, anyway, good enough?
Yep. Yeah.
Good enough.
Great.
Okay,
Great
guys.
job. Great job.
Shoot her in the head. Really? I always feel inadequate to this task. Let me see here. What are we doing?
I mean, this stream did much better. We had a lot more people interested in watching, and so that's
good. Yeah, it's about time.
Yeah. So
okay, we're shooting her in the head. Goodbye out there, and thank you all for showing up. Thank you very much.
Thank you for